Let's go.
All right. Hello, everyone, and welcome to the final session of CEMS 2020. And so we're going to be starting off with a talk by Anna Schapiro with commentary to follow by Michael Mack. All right, Anna, take it away.
Okay, great. Can you see my slides? Yes,
yep.
You can. Okay, awesome. Okay, hi, everybody. Really excited to tell you about some of the things that our new lab at the University of Pennsylvania has been thinking about. So I'm going to start with this very broad question of how does the brain store new information? And I'm going to define two ends of a representational spectrum from localist to distributed representations, which I'm going to illustrate here in terms of their use in neural network models. So in a localist representation, you have some information from the environment that you're trying to encode. There's some set of hidden layers that you might, hidden layer units that you might assign to represent that information. If some new input comes in, you're going to assign a totally different orthogonal set of hidden layer units to represent that information. So when I say localist, I just mean that individual units are only representing one thing in the environment, as opposed to distributed representations where you allow there to be overlap across the things that these individual units will represent. And distributed representations have been very important to the success of neural network models in machine learning applications and in understanding the brain and behavior. They're very good at learning structure and data when they're paired with a good learning algorithm. But there are pros and cons to using distributed representations. So one of the obvious pros of distributed representations is that they make generalization really easy and efficient. So if you're using overlapping representations to represent, for example, all of your memories of seeing birds fly, it makes it very easy to see commonalities with new things that share properties. So if you see a new bird, but you haven't seen that it can fly, because it shares so many features with all these other birds, and you're using this kind of representation, it's very easy to infer hidden properties like it can probably also fly. Whereas that's much harder in a localist representation where you're keeping these related things separate. On the other hand, if you're trying to keep memories separate, like if you're trying to remember that the penguin can swim, but it doesn't fly, even though this very similar looking bird can fly, in this case, it's useful to use localist representations. So they're low interference, and that can be useful depending on the situation. So we have these two different properties that are in tension, and this interference issue is associated with a kind of behavioral difference between these two kinds of representations that I'm going to highlight in this talk, which is that distributed representations are very sensitive to the order of presentation of information. So you can only learn distributed representations when information is interleaved in the environment, whereas localist representations are not sensitive to the order of exposure. So if you've learned about all these birds and then you start learning about some new bird without more exposure to the old birds, what happens is that to the extent that that new bird is similar to the old birds, you tend to overwrite your knowledge of the old birds. That's the classic catastrophic interference problem. Whereas in a localist representation, that doesn't happen because you just assign some new orthogonal set of units to represent that information. So the pros and cons, what does the brain use? Well, probably the brain is using both of these things. And that was the proposal of the complementary learning systems theory, which basically said that the hippocampus is the region that specializes in these pattern-separated localist representations to avoid interference. And then the hippocampus will do the job of interleaving information offline in order to train the neocortex to slowly build up these appropriately situated distributed representations. So the problem with this way of thinking about things is that it takes a very long time to build up those distributed representations, and we can do generalization and statistical learning on a much faster timescale. So we've proposed that actually there are these kind of intermediate representations. So when you're dealing with learning regularities on the timescale of minutes or hours, that there's this intermediate kind of overlap an intermediate learning rate that also exists within the hippocampus that allows you to do that kind of learning. And today I'm going to tell you about some empirical and modeling work that we've been doing to try to test and kind of explore this idea that the hippocampus contains both localist and distributed representations. We've been thinking about these ideas in the context of a neural network model of the hippocampus that has properties that reflect what we know about the actual hippocampus. It has two pathways through these hidden layers of the model that represent dentate, gyrus, CA3, and CA1 subfields of the hippocampus. There's the trisynaptic pathway that runs from entorhinal cortex to dentate to CA3 to CA1. And this is the pathway that implements those really sparse pattern-separated representations that are good at avoiding interference. And the reason that happens is because there's this very sparse fanning connectivity from entorhinal cortex into dentate gyrus that leads inputs, even if they're very similar in entorhinal, to be projected to different populations of neurons in dentate. But there's also this pathway that runs straight from entorhinal cortex to CA1. And CA1 seems to have less extreme pattern separation in its representations. It has less extreme sparsity in the regions that it's connected to. And so we think that this might be where some of these intermediate kinds of representations might live. There's also evidence that the monosynaptic pathway has a slower learning rate than the trisynaptic pathway. So in rodent studies, it's been shown that the monosynaptic pathway is especially important for like incremental learning problems over the course of minutes to hours, whereas the trisynaptic pathway is really crucial for those one-shot kind of learning experiences. So I'm not going to go through these simulations, but I'm just going to quickly tell you that we had shown that the trisynaptic pathway is really good at rapidly learning demarcated episodes, basically. I mean, we already knew this from prior simulations with this model, but if you can think of this as like a word pair learning paradigm, if you tell the model, learn A and B, learn G and H, learn C and D, it can do that really well. And the trisynaptic pathway is responsible for that kind of learning. Whereas if you give the model continuous sequence where it has to learn from statistics over time, that there are these reliably co-occurring segments within the stream, the monosynaptic pathway is the only part of the hippocampus, according to our model, that's capable of doing that kind of learning. So the overlapping representations and the slower learning rate are really well suited to this kind of statistical learning. And we think that might help us explain or understand some of these findings that we had where it seemed like the hippocampus was really important for statistical learning, which on the face of it seems a little bit in tension with its role in episodic memory. Okay, so we have been thinking about the properties of this pathway and what it might be good for. And one of the domains that we've been exploring is category learning. This is also a domain that requires you to often very quickly build up knowledge across episodes over time. And we know that the hippocampus is important for this kind of learning. So we wanted to explore whether the monosynaptic pathway might be doing something special in category learning. So I'm just going to quickly show you a tiny bit of work from Jelena Sucevic, who's been applying this model to different category learning paradigms. I'm just going to show you one of her simulations in a object category learning task with these three categories of satellite objects, where objects in the same category share most of their features. And we've run a few experiments with these objects, and we've found that it looks like the CA1 region of the hippocampus represents this category structure, whereas CA3 and dentate don't seem to show the category representation. So that's some hint from the empirical data that CA1 is involved. In the model, Jelena finds that, so the green is the normal intact network. It's able to learn these categories. This particular measure is actually generalization to novel category exemplars. If you rely only on the monosynaptic pathway, so if you lesion the trisynaptic pathway, the model actually does even better at generalization. Whereas if you lesion the monosynaptic pathway and try to rely just on the trisynaptic pathway, it's really quite bad at that. So the monosynaptic pathway is responsible for generalization in this paradigm, according to the model. And there's actually here an interesting trade-off where getting rid of the trisynaptic pathway makes you even better at that. But if you change the learning problem and you ask, well, how well can the model remember exemplar unique features of these individual objects? The story is the opposite. So it's the trisynaptic pathway that's responsible for that, whereas the monosynaptic pathway is really incapable, at least at this time scale and with this set of parameters, of learning that kind of information. So this suggests that both pathways are involved, but the monosynaptic pathway is really crucial for learning the structure. But it's an interesting additional role of the trisynaptic pathway beyond episodic memory per se, because it's really any kind of situation where you have to do an arbitrary binding of information. So both pathways contribute to category learning. Right, and the hidden layer representations look kind of like what we saw in the fMRI data. So the monosynaptic path we think has these properties that could support learning structure over time. It works for statistical learning. It works for category learning. So have we solved it? Are we done? Is this what's happening, or are there alternatives? Might there be other ways that the hippocampus could be supporting this kind of learning? And to try to get into this question, I'm going to introduce one more paradigm where there have been some really important theories about how the hippocampus might contribute to this learning, which is associative inference, which we've already heard about a few times in the conference. But just to remind you, you learn that A and B go together, you learn that B and C go together, and then there's some kind of test where you have to make the transitive link from A to C. We know from rodent data and from human fMRI data that the hippocampus is important for this task. So I'm going to contrast two strategies for solving associative inference that map onto the two different kinds of representations that I've introduced today in the talk. The first is implemented in this model called Remerge. It's using a localist style representation to solve this task. So the idea is that it forms conjunctive representations of each of the experiences that it has. So it forms a unit for the AB experience, a unit for the BC experience, but during learning doesn't have any contact between those two experiences. There's no overlap between A and C at learning, but it uses recurrent dynamics at test to solve this problem across these separate representations. So the idea is that if you see A at test, you can pull up your AB conjunctive representation, and then that can get you to your B item representation, and then to your BC conjunctive representation, and then out to C. So you can solve this task using recurrent computation at test, but it takes some time and it takes some computation to do that. That's very different than the kind of strategy I've been talking about in the monosynaptic pathway, where you're doing this interleaved learning to build up distributed representations. So there, the idea is that you have items A and C. They start off with some overlap. To the extent that you go back and forth between studying A, B, and BC, over time, there's pressure to increase the amount of overlap because A and C share this B feature in common. So interleaving is important. The order of presentation is important, but if you do that, then it's very easy to link from A to C at test because you have just neurons in common between those two item representations. There's a third very important strategy called integrative encoding, which is kind of a hybrid of these two strategies in a way. The idea is that you learn a item AB, or pair AB, you learn pair BC. As you're learning BC, you pull back up the AB representation and you form some kind of conjoined representation of A, B, and C together. I'm not going to get into this for the sake of time, but for the purposes of the experiments I'm going to tell you about today, integrative encoding we think makes similar predictions to Remerge. So ask me more about that later if you're interested, but I'm going to put it over on the left-hand side. So both the localist and the distributed strategy are available to our neural network model because it has both the trisynaptic pathway and the monosynaptic pathway. So you can see that here where CA1 represents the AC relationship, but dentate does not. Whereas if you allow there to be time for recurrence, you can get the AC relationship to be present using either strategy. So now the question is, like, if you have access to both, why would you use one or the other? Under what conditions might you want to use one or the other? The idea here is that this strategy, the CA1 strategy, should be fast, but susceptible to interference, whereas this strategy should be slow. It takes more time, maybe it's more effortful, but it should be resistant to interference. Okay, so I'm going to tell you about some experiments quickly where we've been testing these ideas about when you might make use of these different strategies. So these are experiments led by Zi Zhou in the lab, and Marley Tandock and Daria Singh are also involved. Okay, so we're going to run an associative inference experiment here where some of the triads are presented in an interleaved order and others are presented in a blocked order. But everything is kind of mixed together. But in the blocked order, you're going to see all the ABs before any of the BCs, whereas in the interleaved order, you go back and forth. After the exposure, we do two tests. The first is a speeded recognition test, where we show people quickly two items they've seen, and we ask them, did you study those two things together during the study phase? And then we do the typical explicit inference test, where we say, there were these transitive links. Here's one object. Which of these other two was transitively related to that first one? We don't expect there to be any difference between the conditions in the explicit inference test, because we think that you can solve that test using either the strategies I just laid out. But we do think that there will be a difference in the speeded recognition test. So the idea here is that if you've built up an overlapping representation of the AAC items, then this speeded recognition test should be difficult. Because if you see an A and C together on a trial, you might get confused and think that you did study those things together, even though you never actually saw them together. So we predict that you should be slower in the interleaved case to say that you didn't see an AC item together, and maybe you would even false alarm and think you had seen them together. So here are the results for the explicit inference test. We don't see any difference between the interleaved and blocked conditions. It's not that they can't do the task. They're really quite good on both interleaved and blocked, but there's no difference between the conditions. In the speeded recognition task, we do see that there's a difference. So people are slower to reject the interleaved ACs, suggesting we think that maybe they've built a different kind of representation. They're also more likely to false alarm to interleaved ACs. This came out again in a new sample of participants, so we think this is reliable. So I just showed you an example of a case where distributed representations seem to hurt performance, but we think that distributed representations are probably useful in many cases. So in the second experiment, we're going to try to show off something that distributed representations should be good at. I see your warning. Thanks, Ken. Which is generalization. So here we're going to do the same training as before, but we're going to then train or teach people about novel properties of some of the objects and test to what extent they generalize those properties to a transitively associated object. And we find that people are more likely to do that generalization in the interleaved case, which we think again is indicative of a distributed representation. And they actually don't do it at all in the blocked case. And then finally, I'll go through this super quickly. We also ran a statistical learning version of the experiment, which we think should really highlight distributed representations even more, because we think that parsing sequences should be very difficult for localist representations. And we find a nice strong effect, again, even in the explicit case. So even in the case where we give people plenty of time to make a judgment, they're much better in the interleaved case. And they actually are not even above chance in the blocked case. Okay, I'm going to very, very quickly in 30 seconds tell you that the results for some simulations with these different models. So I already told you about Remerge and our neural network model of the hippocampus. I'm also going to run, Z ran TCM also on these data. So the reason that we think these set of three models are interesting is because Remerge should behave a lot like the trisynaptic pathway in our model. And we think that TCM might behave a lot like the monosynaptic pathway. It's also using distributed representations. And that's what we find. So Remerge it's totally incapable of showing a difference between between interleaved and blocked presentation. And that can explain our results where we find that there's sometimes no difference between interleaved and blocked in these explicit tests. Whereas TCM, so remerge can't explain the speeded recognition tests. It can never explain a situation where interleaving is better than blocked, whereas TCM is really good at explaining that kind of thing. TCM, in fact, always shows a preference for interleaved over blocked presentation, as you'd expect from a distributed representation. I just want to quickly note this is not due to interleaved ACs being seen closer in time, because both in experiments and in the models, we check foils that are matched for temporal proximity to the target. So it's not about temporal proximity, it's really about learning those representations, whereas our hippocampal neural network model can do both of these things because it has both style representation, although it assumes some ability to control the strength of these two different pathways, which we think we think the MPFC might be involved in that control. Okay, so interleaved learning benefits rapid inference. We think that's consistent with the proposal that the hippocampus might contain these distributed representations in addition to the pattern separated representations. Because I know that Mike is going to talk about this, I'm just going to say that we think this is for a very different reason than in the category learning literature, where trial by trial attentional effects play an important role. We don't think that's what's happening here, so maybe that's an important or an interesting point of discussion for later. And I'll skip that last point. Okay, thank you.
All right. Thanks, Anna, for that fantastic talk. And so now I think we can switch right over to Michael Mack's commentary. Okay, Mike, it seems, I think you might be muted, but now try it out.
All right. Can you hear me?
Sounds good.
All right. Well, thank you. And let me add to the praise to the organizers for translating CEMS to this virtual platform. Has really been a great experience. Now, it's a pleasure to comment on Anna's terrific work. Her research has been an inspiration to me for many years. And the work she was just talking about is particularly exciting. So here in this very brief time that I have, briefer, thanks, Anna. I'm going to talk about three main points. So first up, in the same way that Ida reflected on Greg Cox's model in that first session on Monday, I'd like to place Anna's work along a dimension of computational models that focus on behavior versus models that focus on neural mechanism. As Ida noted, Greg's dynamic model of associative memory is sort of this perfect example of an insightful cognitive model that makes very clear and comprehensive predictions about memory behavior. But using a cognitive model like this to learn about neural mechanism presents sort of a difficult translation problem. So how do you link latent model mechanisms and representations to measures of neural processes? Now, this is not an impossible problem. Many people have successfully taken a cognitive model and used it to provide some insight into the brain. We've seen a lot of this at CEMS over the last few years. I think one shining example of this is work by maybe a CEMS all-star cast of Jim Craigle, Neil Morton, and Sean Paulin. We're able to map trial-by-trial bold signal from MTL cortex and in the hippocampus during free recall to latent mechanisms of the CMR model. And this translation actually serves as compelling evidence that the CMR, a cognitive model developed to account for behavior, is also capturing relevant neural dynamics. Now, at the other end of this continuum is Anna's modeling perspective. So it's based on this rich tradition, the complementary learning systems approach here, and Anna's model is built to first sort of capture the neurobiological interactions within and between subfields of the hippocampus, and then you use the model to simulate learning and memory tests to assess if the relevant behavior actually emerges from these neural dynamics. Now, of course, there's still a translation problem here, but the challenge now is to translate measures of simulated neural activity and representation into meaningful behavior. And Anna's definitely met this challenge. She demonstrates that the hippocampus alone can account for behavior seen in associative inference, statistical learning, and category learning tests. And I think this is an important sort of distinction. By starting with neurobiological mechanism, Anna's sort of uniquely able to provide very compelling computational evidence for the broad role of the hippocampus, not just in memory, but in cognition more generally. So this is my first point, and I'm going to come back to this continuum. But for the next 2 points, I'm going to focus on my favorite act of cognition, category learning. So Anna's focused on the benefits of interleaving learning experiences to better support rapid inference and generalization, something that she sees in behavior and in her model. And this comparison between interleaved and blocked learning experiences or learning sequences is not new. I think it's sort of one of the seminal findings in cognitive psychology. It's been studied extensively in the category learning literature. Here I just have an incomplete list of studies related to this theme that the sequence of learning experiences influences what is learned. There's many more than what I could list here. But sort of the interesting thing about this, we have this giant wealth of data, but theoretical accounts of sequence effects in category learning are fairly sparse. I think the dominant theory, in my view, comes from Paulo Carvalho and Rob Goldstone, who proposed that this is basically an attentional effect. Basically, the nature of a learning sequence changes what features you attend to. Now, with the block sequence, there are many transitions between items within a category, so attention is drawn to those features that are shared by category members. The problem with this is that those shared features may also be shared by members in other categories. So you're not really learning what distinguishes categories. So in contrast, when you interleave category exemplars, it puts the focus on the differences between those categories. So attention is directed to this sort of category diagnostic information. And the predicted outcome then is that interleaving sequences lead to more accurate categorization of novel category items because attention is focused on what actually distinguishes those categories. Now, this theory is fairly intuitive. It makes sense, but it also relies on these comparisons between trials. So it requires this temporal proximity that Anna was just talking about. And when you have these comparisons between trials, it's that comparison, this temporal proximity between these trials, that is actually directly updating feature-based attention. So this is a feature-based attention account of where these effects come from, why you get a benefit for interleaving. However, what Anna has shown us is that these interleaved experiences encourage encoding of distributed representations. And it's these distributed representations in the hippocampus that better support generalization to novel category members. So in other words, it's an emergent property of how the hippocampus builds representations along its two pathways. And it's entirely divorced from this attentional account. So theoretically, this is very, very important. Recently, there's just been a ton of work. A lot of my work has been put into linking cognitive models of category learning to neural processes and representations. And the underlying assumption of all of this work is that attention learning is sort of the central driver in how we build new categories, how we build new concepts. But Anna is showing us that this assumption needs to be reconsidered. These sort of attentional mechanisms might be at play in category learning, they very likely are, but that representations that support category generalization can arise simply from hippocampal encoding functions, certainly requires a significant shift in our understanding of how people learn. Now, a quick little self-advertisement here, sort of to this end, Allie Preston, Brad Love, and I recently outlined kind of a conceptual reconciliation of these two modeling perspectives and something we call the episodes to concepts framework. But certainly sort of further exploring the parallels and differences between cognitive and neural models will lead to important insights like the one that Anna has made here. Now, for my third point, I'd like to briefly highlight some findings from my lab that nicely converge with Anna's theory. In fact, my memory of when Anna and I first talked about this data last fall, I have this sort of clear picture of us giving each other a high five. I don't think that actually happened, but it would have been very appropriate. So Anna just showed us that in the context of category learning, the monosynaptic pathway is sort of key for building these representations that support categorization. And it's the trisynaptic pathway that helps build these localist representations that are key for episode-specific information. But looking at this from a category learning perspective, the very nice prediction that falls out of this is that learning exceptions to categories, like the penguin that Anna had in her introduction, despite the fact that it swims and doesn't fly, it is in fact a bird. This should rely on the functions of the trisynaptic pathway. So in collaboration with Mack Schlichting, we looked at this specific prediction. So we had participants perform a rule plus exception learning task. In this task, I'm not going to go into the details. There's basically category members and a very simple category rule that allow you to categorize most of the stimuli, but then there are also exceptions. And these exceptions are visually more similar to the opposite category. That's what makes them exceptions. So to successfully learn in this task, these exception items need to be distinctly represented. A function that we predict, and I think Anna would agree, is a function of the TSP. And here on the right in the green, you can see sort of end of learning performance for these exception items. And there's a lot of individual variability. Some people do really well with these exceptions. They figured it out. Other people are at chance, and some people they don't get it at all. They're just responding according to that rule. So how can we explain these individual differences? Well, this is where it gets fun. We actually measured the integrity of participants' pathways. So we looked at the TSP and MSP using diffusion-weighted imaging. I'm going to very briefly, I'm not even going to say how we did that, and just say that it's the integrity of the TSP. So the more streamlined counts that you see between CA3 and CA1, that predicts how well people learn the exceptions. On the other hand, or in contrast, MSP did not predict exception learning at all. So consistent with Anna's model, we see this neural evidence implicating the TSP and building the type of localist representations that are key for learning category exceptions. Now to wrap up, what I think is most encouraging about sort of considering Anna's work in light of all of the related cognitive models out there is that we have both sides of this continuum that are working towards the other side. And this gives us a real opportunity to bridge levels of analysis. And the really helpful thing here is that we can do this with just the models themselves. We can run model simulations with changes in neural mechanism and see how that leads to changes in cognitive mechanism and vice versa. And that's going to lead to important insights. And this, of course, begs the question of who needs participants anymore? We can do this all from the comfort of our home offices, which is perhaps good news as we wait out the pandemic.
All right. Thank you very much for that fantastic commentary, Mike. So I thought I'd give Anna a chance to quickly, maybe in just a minute or two, respond to Mike's commentary, and then there's a question.
I don't think I have I agree with everything Mike just said, and I am very excited about that data. So I'm happy to go to questions. We have an interesting one from Q here. Okay, fantastic. Yeah, so let's see. Can we actually? I'll just read it in the interest of time. So Ki-Hong Liu asked, he said, super cool experiment, Anna. Do you think this training experience can explain how information is represented in the brain in general? The brain seems to represent many things in a distributed fashion, but some information seems to have a dedicated region, although they might not be completely localist, such as the representation of faces. Is it partly because humans got trained with faces in a block setting over a lifespan or over a generation? So, okay, a couple things to say. I mean, I think that distributed representations are the rule, not the exception. So, like, the brain uses distributed representations, and the dentate gyrus region of the hippocampus is fighting as hard as it possibly can, using very strange, different kinds of connectivity to resist that natural tendency of the brain to use distributed representation. So, I tend to think everything is distributed. The magic of this monosynaptic pathway is just that it can learn more quickly than most of the brain. So I think that that's really exciting from the perspective of testing ideas about distributed representations, because we don't have to wait for developmental timescales or perceptual learning timescales to test these ideas. They can happen within an experimental session. I don't think that the nature of the learning experience leads to different kinds of representations necessarily. So like if a brain region is set up to do localist representations or distributed representations, it's not that blocked learning will cause there to be localist representations. That's not how I think about it. Although maybe over the course of evolution, like you're saying, that could happen. And maybe that's what happened in the dentate gyrus with its specialized connectivity. I think that face processing, well, I'm not an expert, so I don't want to speak to this too much, but I think that it does have a lot of properties of distributed representations, but for whatever reason, things have become relatively more categorical. There's been pressure, whether that's through training or through evolution or some combination, to kind of keep the types of features that faces are involved with in separate processing streams. But I would say, I think of most of the brain as distributed, and certainly the ventral temporal cortex.
Great. So we have another question from Kevin Himberger, which I'll just read. So both great talks. I think Anna mentioned the MPFC potentially up or down regulating the MSP versus TSP. Is this correct? And if so, could you discuss potential mechanisms?
Yes, that is what we are thinking about. I probably cannot tell you something very useful about potential mechanisms. I mean, I don't think there's here's what we know. We know that CA1 is connected directly to MPFC in humans. That's really cool. We also know from rodent experiments that the MPFC seems to have some control over what's happening in CA1. But the idea that the MPFC is routing information differently through the pathways, I'm not so sure if there's evidence for that, although there's plenty of evidence that we can route information differently through the pathways. So we know that, you can be like Catherine Duncan's work, you can be more in an encoding or retrieval state, that we think that routes information differently, or levels of acetylcholine can route information differently. The theta cycle does, but we'd probably be interested in a longer time scale than that. So the idea that the MPFC could bias the strength of the two pathways through its action on CA1 seems plausible to me, but I think it's at this point it's just an idea.
Okay, so I'll ask one last question, which is from Rachel Diana, and it's: can cognitive strategies like attention modify neural pathways and computations? And if so, is it the case that even a behavioral finding that emerges from neural function needs a cognitive explanation?
That's for Mike.
Okay, Mike, take it.
Is it necessary? I would say absolutely, because we like to understand things at different levels of analysis, different levels of abstraction. And there's a whole rich literature of cognitive models that talk about attention and specify the type of impact attention has when we learn. And so understanding that both there's a neural mechanism for it and that these behaviors emerge from that neural mechanism gives us a lot of insight into that whole rich history of effects that have been associated with those models. So it might make us pause and say, maybe this isn't due to an attentional mechanism and something that emerges out of the encoding that we see in, say, the hippocampus. But that's an important sort of reflection that we can do on the history of all of these effects.
All right. Thank you very much for the fantastic talk and discussion. So our next presentation is by Nick Diamond from the University of Pennsylvania. So I think we can switch over the videos and hopefully get Nick up. All right, there's Nick.
Hello.
We can hear you, so just share your screen and then you can launch. All right, great.
Okay. Great. Okay, so hello, everybody. I'm a postdoc in the Kahana lab, and today I'm going to tell you about some of my PhD work with Brian Levine at the University of Toronto. So most of us can cast our mind back to many specific events spanning our life history, and these are the basic units of episodic memory outside of the laboratory. Now, these events are not just discrete slices of time or points in memory space, but they're embedded in extended episodes, they're trajectories. And like word lists, the way that these episodes unfurl in our minds can tell us a lot about the retrieval process. However, these episodes have been little explored, or sorry, the temporal structure of real-world episodes has been little explored because usually these episodes are unverifiable. As experimenters, we have no control over the encoding experience. So one solution to this problem is to design controlled real-world encoding experiences. So this is what I did in my graduate work. For example, here's an audio-guided walking tour of the artwork in Baycrest Hospital. So people encounter a number of strange and interesting objects in a fixed sequence. For instance, they have to find this ceramic chef cookie jar in a gift shop. And this event takes about half an hour and covers quite a distance. Now, after 2 days or a week, we had younger and older adults freely recall their experience, narrative style, just like you would with any personal experience. And because we had control, we can apply tools from the wordless learning literature to look at recall dynamics. And as you might expect, or maybe not, we see the hallmarks of temporal context reinstatement. I'm saying temporal even though time and space are fully confounded here. But the point is we see quite striking degrees of contiguity and forward asymmetry, even after several days or a week. Like in wordless studies, older adults in the dark gray green line here reinstate context less. We also found that these dynamics related to the types and the quantities of details that people recall. So the point here is that sequence structure shapes how we recall real-world episodes, even quite a while after the encoded episode. But what about years-old memories? We routinely see these in autobiographical memory experiments. All of us can recall probably many years-old memories, but we don't really know anything about the structure inside of these memories. And what is the neural basis of retrieving within episode sequence structure at this time scale? So this brings me to a second controlled real-world encoding experience, and this is the brain exhibit. So this was a real museum exhibit about the brain at the Ontario Science Centre in Toronto. The fact that it's about the brain actually is totally incidental to our questions. It just makes this all extremely meta. So anyhow, people encountered a number of objects in an order that was determined by the track-like layout of the exhibit, also by a scavenger hunt style pamphlet that we gave people. And after a delay of 3 months, we did an initial behavioral experiment, which I'll describe in a moment. So this involved old, new, and order discrimination. And we published this a couple years ago. It was an aging study. And we followed up with the younger adults in this study, 18 to 22 months later, and did essentially the same task while these subjects were undergoing fMRI scanning. So this is roughly 2 years after the initial experience, a one-shot experience. So in this task, subjects were presented with pairs of pictures. They could be intact if they were presented in their encoded order, going from left to right. They were reordered if they were presented in the incorrect order, going from left to right. We also manipulated the distance between the pairs. So near pairs were adjacent in the exhibit. Far pairs had 2 intervening items. Now, to contrast memory for order with mere recognition memory, we also got lure photos from another exhibit about the brain, which happened to be in the Franklin Institute in Philadelphia. This was before I moved here for my postdoc. And through a pilot experiment, we identified photos that were the most plausible, the most confusable with our target photos. So these are perceptually and conceptually similar to our targets. And to be clear, on each trial, people saw a pair of pictures, and they responded intact, reordered, or new. Okay, so let's look at the behavior in the scanner. Remember, this is 2 years post-encoding. So I'm going to show you performance for each of our trial types. And chance is 33% because there were 3 response options. And the main takeaway, maybe the main takeaway for this whole talk, is that people can do this task. So not only can they discriminate targets from lures, but they can also reconstruct order at a pretty fine grain, of course, given appropriate retrieval cues. Now, there are 2 patterns in these data I want to highlight. One is an order effect. So intact pairs elicit greater accuracy than reordered pairs. This is consistent with the encoding specificity principle. If you present retrieval cues in a manner that matches the encoding context, you're going to help retrieval. There's also a weak but significant distance effect, where far pairs elicit greater accuracy than near pairs, and this is consistent with many previous studies. Okay, so here I'm just briefly showing you performance on that initial test, that 3-month behavioral test. I'm showing you the means for these subjects in red triangles. The point is that, of course, memory got quite a bit worse from 3 months to 2 years, mostly for the new trials, but I'm not going to dwell on this now. Okay, so what should we expect in the brain? Of course, this study is partly exploratory, given some of the unusual aspects about the design, and we expect that quite a distributed brain network should be involved in this task. But naturally, we were curious about the hippocampus. We know that hippocampal dynamics are critical for sequential organization and memory. The hippocampus in previous fMRI studies is sensitive to distances and times between familiar locations and events. And lastly, the role of the hippocampus in remote memories at this years-long timescale has been debated for decades. Recent evidence suggests that hippocampal activity might reflect memory vividness rather than memory oldness per se. And vividness itself is thought to be related to spatiotemporal context reinstatement, which we can measure objectively here with our recognition task. So I analyzed the fMRI data using PLS, which Signe Sheldon explained really nicely a couple days ago. Very briefly, it's a multivariate data reduction technique. The input to the analysis is a brain matrix and a task design matrix. Using singular value decomposition, PLS extracts latent variables that are always, they always manifest as a contrast. So what you get is contrasting spatial patterns in the brain, which reflect some contrast between your conditions. And the key difference, or the key advantage relative to a univariate design, is that you're getting a multivariate pattern in one step, rather than a series of GLMs at every voxel. Okay, so I'm going to show you two contrasts. The first is ignoring order. So we're just looking at hits versus false alarms. And when I say hits, I mean calling any intact or reordered pair intact or reordered. And false alarms would be calling any new pair intact or reordered. So just old and new. BSR stands for bootstrap ratio and can be interpreted like z-scores. So here's what we find. The warm voxels are more coactive for hits. The cool voxels are more coactive for false alarms. And when people correctly recognize the photos from the exhibit, we see a cluster in the right anterior medial temporal lobe in what I believe is the perirhinal cortex, previously associated with discriminating old from similar new information. And then beyond that, we see the standard cortical recollection network or default mode network or autobiographical memory network. So the medial prefrontal cortex, medial parietal cortices, and lateral parietal cortices. So this is consistent, doing a bit of reverse inference. This is consistent with sort of recollecting past episodes, generally speaking. However, of course, we wanted to zoom in on memory for order. So here, I'm contrasting correct order trials versus incorrect order trials, all within those old hits. So anytime you called an intact pair intact or a reordered pair reordered versus calling an intact pair reordered or a reordered pair intact. So here we see two significant clusters.
1 minute.
Got it, thanks, Ken. One in the sort of mid to posterior region and one right at the posterior end of the hippocampus. And speculatively, this is consistent with the idea that there's a gradient of granularity along the hippocampus where more posterior regions represent more fine-grained spatial and temporal information. Of course, here people are reconstructing sequence at a very fine grain within a single episode. Previous studies looking at spatial and temporal relations across events found more anterior activity. Now, as I mentioned, PLS is blind to the direction of the neural signal. It's just pulling out contrast. So I wanted to look at the raw signal change just for descriptive purposes within these clusters. You can see in both cases, only when people correctly retrieve the order do you see an above baseline increase in hippocampal activity. Okay, so wrapping up, sequence structure shapes retrieval of one-shot real-world events, even after very long delays. And I think this reflects some kind of retrieval of context. And speculatively, again, we see a time-invariant role of the hippocampus in recovering sequence information. Of course, we only scan people at one time, so we can't speak to neural changes over time, but that's something we may do in future work. So I want to thank Brian and the Levine Lab, and also my current supervisor, Mike Coutanche, and my current lab mates, and of course, CEMS organizers for making a really fun and great conference under these circumstances. Thanks.
All right, thanks for the fantastic talk, Nick. Let's see, while I guess we give people a second to type in questions, I have a question. I just wanted to invite you to talk more broadly on how you think your results relate to the findings from more stripped down list learning paradigms, right? You've got this incredibly interesting real world event. Do you see this as confirmation of principles that have been found in wordless learning experiments, or is there anything different going on process-wise here that you think isn't happening in the list learning studies?
Great question. I see between the two options, I lean more towards confirmation. So I think in this study, also in the recall study I alluded to at the start, we're seeing an extension of really well-studied principles to this more naturalistic context. I do think it has some features of these tasks have bearing on theories of what's going on here. So in similar studies done in the lab where you have two pictures or two items and you're judging which one came before the other, this is often thought to reflect a judgment of relative recency. So you might ask which item is closer to now, which one is stronger in memory. Now, I don't think that can explain performance after 2 years, because 2 years and 1 minute is not so different from 2 years and 2 minutes. So I think this shows that people can recover, people can do this task. They must be jumping back in time rather than just making judgments of recency. So that's one example of, a consequential difference.
Thanks, that's great. So we have one question in the queue from Alex Barnett, which I'll just read, which is, was there an effect of the temporal spacing on brain activity when making order judgments?
Yes, so I did look at the difference between near and far pairs, which is what I think Alex is alluding to. Basically, we see, I'm not going to pull up my slides now, but we see a large medial prefrontal cluster and large clusters in the caudate bilaterally. I don't want to speculate about this too much, but that's what we find. No hippocampus.
Okay. Let's see. I think in the interest of time, we need to move on to the next talk, so we can all thank Nick for that fantastic presentation. And so the final short talk of CEMS will be from Marc Coutanche, and so hopefully Marc can get on stage and present to us. There's Marc.
Hello. Okay.
All right. We can hear you, which is step one.
Great. Let's see how this goes.
Now we can see your slides, so you're good to go.
Okay, excellent. Great. Thanks, everyone. And also, I'd like to echo the thanks to the organizers, not just for putting this together, but for making it financially accessible to students. I think that's really cool, and I appreciate it. I'm sure they do too. So what I'll be talking about today is some work from my lab, which relates to quite a few talks and presentations that we've seen, actually, which is on recalling the when, where, and what of naturalistic episodes. So let me see if this advances. Great. Okay, so one of the benefits, the curses and the benefits, I guess, of being the last speaker is that a lot of what I've said, what I have, or what I'll be introducing has already been mentioned. So, but I still need to sort of set up the study I'll be talking about. So we'll go over it a little quickly. As we know, naturalistic or dynamic episodes have got high dimensional facets, right, of memory, as well as the item that's featured. There's a spatial context and temporal information. And that's the kind of information that's not often, well, certainly temporal information isn't present at all in static images or words or things like that. And this presence of the spatial and temporal dimensions is more than just one additional dimension, very likely influences how information is grouped together and understood. And we've seen a lot of work here and in many past years by people here kind of digging into this. And sure enough, we find that if you kind of control for information that you're giving people, it's more likely to be remembered when it's the same information embedded in a dynamic context than in a static image. So there are extra things going on that enable things to be learned in a superior or let's say different way, which I'll be investigating here, when it's embedded within a naturalistic episode. So the main, I've got kind of two parts to this, all around one experiment. And the aim of this experiment was to ask how different factors, different kind of influences that we know have an impact on, learning and memory of static images and words, how these different factors influence recall of naturalistic episodes. So I'll first introduce what these influences are. So first of all is prior familiarity. Okay, so having familiarity with an item will often, to speak broadly, affect or improve episodic memory strength. We've shown this before, as have many others. So it's a pretty robust finding. And also, it can affect its character. So it's not just about being familiar with something, meaning you are more likely to learn related information, but actually it can affect, for example, in work last year, how whether you form bidirectional or unidirectional associations and various other things. So having prior familiarity matters. The second factor I'll be addressing in this study is sleep between encoding and retrieval. Again, we've heard some of related kind of concepts here at the conference, so I won't belabor that. But I'll just note that when you dig into some of the effects that sleep has on memory, the particular types of information that are benefited, there's a real emphasis often on contextual details, right, or spatial relations.
Hey, Mark, we're getting a little bit of a buzz in your audio. I don't know if there's anything.
Okay. Is it an echo or a buzz?
It's a more echo.
Okay. I tell you what I'll do. I'm going to mute. I can't hear you. So is this better or is it the same? I can only see you.
Same.
Hang on. Testing. How is this? That's just how it sounds.
Still.
Okay.
Yeah.
So it's not to do with muting the tab.
Is there an extra tab open or?
Only one. It could be that the laptop's running a bit hot right now. Let me move this away. How is this? Not really.
I think it probably is with the Crowdcast software. I think, if you're not, are you using AirPods or anything? No, I don't.
No.
See them. Okay. Oh wait, it sounds better now all of a sudden. Try it again.
Testing?
Yeah,
no
good.
AirPods
You're good.
here?
Go for it.
Oh, okay. I won't touch anything.
Yeah.
Great. Okay. Where was I? Maybe I'll do the mic thing. It's good now. Great. So where was I? Oh yeah. So sleep, we know, it has an impact on contextual details, spatial relations, other things too. I just want to point out that there's that link. And finally, the third thing is trait differences. So there's some work showing that, especially in autobiographical memory world, that, okay, thanks, talk fast. So self-reported tendency to remember episodes is measured to certain memory performance. So kind of questionnaires, when people respond highly on when I remember events, I'll remember a lot of details. When things like that, they're more likely to remember certain things. So it's a survey of autobiographical memory. So we wanted to look at how these factors influence different facets of recall. In the same way that one might look at recognition, and recall in an experiment, we wanted to look at cued recall from these naturalistic episodes or videos, free recall, and recombined recall. So I'll mention in a second what this is, but it's very analogous to what Dan was talking about. Oh, okay, thanks, Sean. Prior familiarity. So in terms of hypotheses about these different forms of recall, we kind of suggested or thought that prior familiarity would particularly help cued recall. The reason we thought this was that familiarity is associated with improved associative memory, especially when there's interference present. Now, you can get interference, of course, with free recall and cued recall, but our reasoning here was that when someone is just given a free recall task and are able to, say whatever they want, they're more likely to bring up stronger memories, okay, and follow down a kind of a stronger memory route. Because of this, we thought that the cued recall, where we are asking a question they have no anticipation of, they're more likely to be perhaps vulnerable to interference, and so we wanted prior familiarity to have a role on cued recall more than these other types. We hypothesized that it would aid free recall, the sort of sequential replay during sleep of neuronal patterns that occur during encoding may facilitate particularly recall in the sense that there's this temporal and spatial relationship that is present both neuronally and also behaviorally as someone is going down the free recall list. And finally, and this was a little bit more speculative, but these trait differences, we really drew quite heavily on some of Dan's work. So it was fantastic seeing him talk about this here, in particular, these episodic inductions and looking at and seeing these findings where you induce some kind of episodic thinking and it improves this kind of construction or recombination. And so we thought that, well, the episodic trait, although it's not an induction per se, could almost be like a self-induction or just, perhaps more susceptible, if you like, to having induced episodic memory. So we had an idea that this trait differences might be related to recombined recall, but also this was pretty exploratory. We didn't have any strong ideas. Well, we had 115 participants. They came in, they viewed 24 silent nature videos. These featured 6 generally unfamiliar animals, kind of rare animals. Each of these videos lasted 45 seconds. So I won't play them all here, but to give you kind of an idea of what's happening, these are the kind of videos that they would see. These animals doing various things. I think there might be a little bit of a lag here, but you can hopefully see that there are, this a handsome guy here going about his business. And people were asked later, what was happening in terms of free recall, recombined, and cued recall. And the cover task they were given was to track their interest during the video. We didn't actually use that. That was just to make sure that they don't necessarily directly suspect that there's a memory test coming. Okay. So let's see. Great. So after the encoding, they actually came in and were given 3 of the animals to immediately retrieve. And then they went away for either 12 hours or 24 hours. So we had a design when they came in the morning and then came back in the evening, or came in at the beginning in the evening, came back in the morning, and 24-hour groups for AM to AM and PM to PM, so that we could both look at the effect of sleep, but also try and dissociate that from time. So it's kind of a fairly common design for looking at the effect of sleep over, say, wakefulness. And we counterbalance this across participants so that people would only be retrieving information from 3 of the animals, right, during immediate recall, and then the other 3 during delayed recall. So free recall was, can you tell me what you saw in the videos? Pretty open-ended cue, or I shouldn't say cue, but prompt. Recombined recall was, if you had to describe a day in the life of this animal, what would you say? So participants weren't given a day. They weren't given the information of when the animal wakes up and so on. So we asked them effectively to kind of put together this information in a new way. And we had cued recall questions. So for example, what did the animal do while it yawned? There are actually 16 of these questions for each of the animals. And we had questions that directly tested temporal, spatial, and attributes. So that's the when, where, and what that's occurring here. I'll also add that although cued recall allowed us to target these, we went into the free recall and the recombined recall transcripts and parsed and coded among multiple people whether people were recalling a temporal, a spatial, or an attribute. In fact, actually, because of the number of videos and the number of questions and so on, we ended up with over 10,000 transcripts. It seemed like a good idea at the time. That's all I'll say on that. Okay, so let's just jump into the results. So prior familiarity was strongly linked with cued recall. And when we broke this up into the different facets of cued recall questions, we found that temporal questions, what happened after the bird landed, for example, were particularly related to familiarity. Spatial was trending, and attributes were not. Now, just to clarify, that might surprise you, but actually we chose the attributes to be properties of the animal that were not directly related to knowledge. So it would be, right, it's, you had to be familiar with the episode in order to answer correctly. Maybe it was the particular pattern of the spots or particular shade or something like that. So we saw temporal information was particularly benefited by higher...
1 minute, Mark.
How many?
1.
Oh boy, okay. But not free or recombined. When we look at sleep, we found that actually it was free recall that was impacted, and wakefulness led to a drop across all three, but sleep seemed to protect that. And the trait differences, I'll just skip over this, but it was a little bit messy, but we saw as people were higher in some of the traits, they showed higher free recall performance and recombined recall performance. So I will skip this. Oh, interestingly, if you enjoy the cover of the August Learning and Memory, if people look at covers of journals anymore, that was by one of our co-authors, sort of reflecting how the familiarity with an animal can make up or support particular episodes. And I won't go into this last part because I got sort of derailed, but I will just say that we looked into the free recall transcripts further and used LSA to look at how coherent the words were. The sentences were, and we found that one big impact was that sleep caused a reduction in semantic coherence. So from sentence to sentence, were less likely to be related to each other if someone had slept. Okay, I got some interpretations there.
Don't rush through the interpretations. You can read them unless there are lots of slides following this.
Okay, yeah, I'm right near the end here anyway.
Yeah.
And so this last piece here where we're seeing this, as a reminder, we're seeing this drop in semantic coherence with sleep in free recall. This drop with sleep across both sequential sentences and overall is kind of consistent with this idea that sleep counters trace decay by forming these non-overlapping representations. And it seems to come at the cost of semantic coherence. I think this kind of relates kind of nicely to some work from Rebecca Cutler and colleagues who showed that healthy controls generated semantic features to concepts that were less similar to the target than did patients with hippocampal damage. We're showing here that sleep leads to less similar, conceptual relationships. And then Sarah Solomon and Anna Schapiro actually commented upon this and suggested the hippocampus might contribute this connective code to semantic search, allowing quick access to distant features. Well, our findings may suggest, it's early, we want to do some more work, but I would say, there's some suggestion that perhaps sleep will increase accessibility to these features of the episodes that are located further in semantic space. So I just want to thank you, my lab, and the people who worked on these projects. Thanks.
All right. Thanks, Mark, for that great talk, for being a good sport with the strange buzzing. So let's see if we have any new questions coming up for this talk. So let's see. Looks like these were mixed in from the last talk. I can ask a question while this is getting sorted out. I just wanted to get a little bit more clear on the decreased coherence after sleep and recall and how that was measured and how, I guess, I think that you sort of zoomed through that. It seems like an interesting finding, and I didn't quite get what it was.
Thanks for asking. Yeah, happy to. So, what we did is we used LSA, but instead of a word level kind of measurements that you might typically get, we used sentence level coherence. And this kind of sentence level metric where you effectively in vector space look at the distance between like an average of the words in a sentence and other sentences, has been linked to things like comprehensibility, right, of passages and so on. And for something like a free recall task of a naturalistic episode, it seems a little more ecologically valid and useful compared to kind of a word by word LSA analysis. So that's what we did there. And the drop, with sleep really points to the after sleep, people are effectively the sentences, the coherence, the relationship between them become more distant from one sentence to the other.
Right. And so was that unconfounded with time of day?
Yeah, we put all of that in the model, including things like number of words, number of sentences. There were some time of day effects in some of the earlier results. We also, though, once we controlled for that, we also found the sleep effects, but there were some time of day things happening, especially people in the morning doing better.
Right. Thanks, Mark. Okay, so I think that with that, we can call this session to a close. I wanted, so there's still a poster session that I hope everyone will attend, as there are many fantastic posters left to present. I did want to close by thanking again Georgia Riley and Connor Keene, who behind the scenes made sure that all of this very novel and strange and sort of challenging virtual conferencing things went off smoothly. So maybe a virtual round of applause for Georgia and Connor. And I also wanted to thank the committee, so Sean Pollan, Maureen Ritchie, and Lynn Lonis, who I think had absolutely no idea what they certainly they had no idea what they were signing up for when they generously agreed to be on the committee, and I think really successfully charted a course into very unknown waters, and I think this has been a really cool experience. I think we've learned a lot, and even if we fingers crossed that we can do this in person, there may be some elements from this that we can carry over to future in-person versions of SEM that would be really nice. So thank you all. Enjoy the poster session, and we will be in touch about next year when the time comes, and hopefully the Earth is still around. Okay, take care, everyone.